Gross. Yeah.
I know. People joke about it though because it's goofy.
Yeah.
Well, I think they make some Jolly Ranchers too.
Yeah.
Drink. I mean, that's my one team. Thank you for coming to my.
I like your Pokemon team, Grace. Very creative.
I know.
Yeah.
I forgot to turn these in
Oh,
all day
because they're good.
it wasn't working. You want me inside?
It's
not the right house.
It should be on the other side. You can like turn it over. Yeah. You can want to pass it around.
Yeah, it is.
Okay, we got Mackenzie and Hannah here today, right?
That's not even.
Did you guys make any edits in the past couple days? Okay.
Whenever I turned my head, I immediately made an edit because I wrote down something dumb and then I really was dumb.
I did with an apology for.
Please, yeah, don't worry about it, please. Yeah.
The only thing that it did change was the 2.5.
Okay. Greater, where is it? There it is. 'Cause I want to be sure I pull up you guys' like current ones. So give me a second to get that together and then we'll get started. So today we have what's that? You got 20, you got full points, right? Yeah. Don't worry about it. Yeah, you're good.
So don't worry about it.
If you got 20, it was 25 out of 25, right? Don't worry about it. Okay. While I'm getting all this set up, does anybody have any questions about anything today?
Do we have to do all that? I'm joking.
We can rely on it every day from Grace. We can rely on that. The answer is still yes.
Yeah.
Yeah, it's okay. You're fine. Monday, Wednesday. Oh, that's just the one that says that exam is still tomorrow. That's right.
Reading are unknown.
Yes, we are. So you should have, by the end of today, all of your results. Everybody should have all of their results today to make your guesses. I'm not saying you have to make your guesses today, but the odds of me getting to, like, if everybody waits until Sunday to submit your guesses, and the odds of me getting through to everybody and telling you why your guess is wrong or what test went wrong, because not most, I'd say probably half of the time, you're going to make a wrong guess because something about your bacteria didn't behave, or somebody misinoculated something, like it just didn't grow one of those tumors or whatever. So we don't know till we make our guesses. And I have to make sure you guys are using the key right and not just like, because you're gonna, on the exam, you will have one of those, you know, your little chart that has whether your gram-positive was positive or negative for all these things, the big chart, like the other thing on the table. That, yes, that chart with your gram-positive, did it get positive or negative for all of these tests? Did your gram-negative get positive or negative for all these tests? Was it A over K? Was it gas? Was it whatever? That chart. You'll be given one of those charts and you'll be given a dichotomous key. And I will have you guys tell me what the organism is based on just those two things. So that will be 2 questions on the exam like that. It'll be 49 and 50. It's always the last 2 I was in for the end. It should be easy, right? You should be able to look at a chart and say, this was positive for this, and look at your dichotomous key that I give you, which may not be the same as the one that you have. So to be able to follow that chart and see where your organism is. Number one thing about using a dichotomous key, you always start from the beginning. So if you're like looking at yours and you're like, oh, mine was positive for indole, you don't go and find on the chart where indole is and then start from there. you go from gram negative to does it make gas and glucose or not to whatever the next steps are, and you always follow them stepwise. Because there's a reason why it's set up the way that it is. So keep that in mind. Some people try to make guesses because like, oh, this shows blah, blah, blah thing. This must be my organism. I also have people submitting guesses yesterday for their gram negatives that require the answer to the nitrate test. Don't, you ain't jumping ahead. We still got to do that test. You ain't got results for it. I'm not going to tell you. I'm going to say we have not done that test yet. Today, we'll have all of our results, so that shouldn't be an issue, but I've had 3 people submit their guesses for their gram negative, and that gram negative that they're guessing required that it be negative for nitrate reduction, which we have nobody in the entire semester has got a result for that yet. So, nope. Or they'll be guessing, well, my gram positive is Staph aureus. How do you know that? Is it catalase positive or negative? That's the first thing in the cocci category. If you have a cocci, does it make catalase? How do you know? We haven't done that test yet. So that's all I'm gonna say. So I just wanted to make that statement before we let me pull up everybody's stuff. Got it, acquired. All right, so no other questions. Okay, so we've got Kuru, we've got polio.
I like seeing there's a student named Kuru. Like, there's a student named Kuru?
Unfortunate naming.
That would be an unfortunate name.
We've got rabies.
Really?
Yeah.
Well, maybe we could do that. No, I think it's your.
Is that your screen?
Yeah, privacy screen. Yeah.
Awesome.
Girl. And, we felt like the. Yeah.
Okay.
It's really not, it's like 77 right now, but I feel like it's like 110. Oh, no, it's at 92.
You're hilarious.
Here it is. Okay. And mine. So we'll have Stephanie go first, since hers is tied into the skin. We touched a little bit on ringworm. That's what her talk's going to be about. So she's going to give her presentation first, and then we'll just run through everybody that's on that list on you guys' schedule today. My online folks, for Mackenzie and Hannah, you guys are going to go last. Just FYI. It doesn't really matter. Kind of, I guess I'll have Hannah go first and then Mackenzie, because your chapter, Hannah's chapter comes before Mackenzie's. I don't think it matters, but whatever. That's how we're going to do it. So let me pull this up. Downloads and 1, 2, 3, 4, 5. Okay. Okay, let's pull up Stephanie. Is it coming up? Are we working today? There it is. Forget this. Got this recording. All right, Stephanie.
My life for like a dollar. Okay, so if I forget something, I apologize. I'm actually really nervous. So mine is on ringworm, and another name for ringworm is dermophytosis. Ringworm is definitely a highly contagious type of fungus or fungi. It's usually on the skin, hair, and nails. It is highly contagious if you get one. And the, a lot of people, so you can come in contact it, human to human contact. If you're in sports, you can get it through sport, especially for wrestlers. Wrestlers are very, they definitely get ringworms a lot. I have kids that wrestle and we have ringworms all the time. And no matter how clean you are, no matter how you disinfect it, showers, everything, you still come up with ringworms, and it's animal to human objects. So if you take a shower, don't reuse your towels. If your clothes, make sure you wash your clothes, don't rewash them, because, or your bedding, always wash your bedding. Treatments for ringworm is with different types of topicals. So I just went blank and I know these ones. So the chlormidazole is a topical. The miconazole is a topical that usually is used. If they're really bad, I can't remember the name of the antibiotic, but there is an antibiotic that they will give you for ringworms, but they don't usually give you that antibiotic. Usually they try you with the topicals first. And basically the pathogen is a fungi, and it will break down the keratin in your skin, and it stays topical, but it will break down the keratin of the skin, hair, and nails. And the signs of a ringworm is it's usually round, red, definitely itchy. I've noticed when like my kids get a ringworm, it starts out looking like a pimple. And then they're like, oh, it itches. And I'm like, don't touch it. Do not touch it. And I'll try to put some medicine on it to keep it from spreading. But it definitely does spread. The worst case I've ever seen in ringworms is there was a little kid that had it from head to toe. And it was and you were like, don't come around me, please. But definitely, and the way that people can tell whether it's a ringworm, most is by visual. They can tell, but they can do a scrape test to see. They can do also, oh, I'm sorry. They can do a culture or a PCR or even an immunofluorescent test to determine what kind of ringworm it is. And that's it. And this form that I gave you tells all about the ringworm, the treatments, and the pathogen, and how you can prevent them and keep from getting them. Just make sure you wash and shower and clean all your stuff. And that's it.
Oh, who? All right, Marissa, are we ready? Oh, I will say too, Leanne, we have a lot of people going today, so I'll probably have you go next week sometime, probably Monday. Just to let you know, yeah.
Wait, how do I move it before you leave?
Oh,
I don't know how to use it. Should be.
That's my fault. There you go. Thank you.
Okay, so today I'm going to be talking about Kuru. So a little bit of the basics of Kuru. Kuru is a rare and fatal brain disorder that occurred between the 1950s and 60s among the Ford people in the highlands of New Guinea, which is just a tribe in New Guinea. The disease was a result of practice of ritualistic cannibalism among the Ford, where they prepared deceased relatives, and it was basically, they saw it as a way of freeing their spirit, so it was more of a respectful ritual than anything. It does not have a scientific name, but it does fall under the TSE category, which is essentially a prion disease. So moving on with the virulence factors, like I said, it is caused by prions, which are abnormal proteins that form in the brain that also create holes. And the holes are basically a pretty telltale sign of Kuru and that degenerative brain disease that it is. And then proteins do not have genetic material, but even without that material, it can still spread to a new host and that disease will continue. And the mode of transmission, even though it's highly infectious, you're really only going to get it if you eat the brain of someone infected or their tissue, or if you come in contact with open sores or wounds. You're not going to get it by like someone coughing on you that has Kuru, or like if someone touched a doorknob that has Kuru, then you touched it. You're not going to get it. And the risk factors, once again, there really aren't many risk factors. It's rare unless you literally come in contact with Kuru, you're not going to get Kuru. But it does have the codon 29 gene, which if you have the gene, you're basically just more susceptible to any TSD, not necessarily Kuru. However, Kuru was more commonly found in children and women. They're not more susceptible. They were just the ones who typically ate the brain during these rituals. Don't know why. Wish I could have told you why, but I guess just men didn't eat it as much. Symptoms and signs. Really, once you got it, within 6 to 24 months, symptoms have basically taken over and you're to the point where you're going to pass away from the disease. It affects the cerebellum, so you basically lose control of your body really immediately, but as it worsens, you will lose the ability to swallow, you won't have control of your bladder, you won't be able to speak, or you won't want to speak, and you really won't be aware of your surroundings. This is where you do start to, or you would start to get the chronic bed sores, which do become infected, and that's where it would continue to spread if you came in contact with those sores. But what I thought was really interesting is that Kuru can be undetectable for 5 to 50 years. So a lot of people probably went through these rituals and died not even knowing they had Kuru. And then, someone else ate their brain and it just continued and you wouldn't even know. Diagnostic methods. Like I said, since it's so rare and only really happen in one part of the world, there's really not a method to like determine it. But you can go through a couple other methods to rule out other diagnoses. Like you can do neurological exams just to understand, what's going on with the body and the muscle jerks that are related to Kuru. You can also do blood tests to rule out other things that kind of look like Kuru. But like I said, none of these alone can determine the presence of Kuru. But actually, after postmortem, they can evaluate your central nervous system, and that can confirm it. But that would only be after death. And then care and treatment, like I said, once you got it, you got it. You can really only find ways to relieve it. Pain meds and physical therapy can kind of help make it less horrible. But once you're to the point where you show symptoms, it's just a matter of time. And that is really all. I just wanted to add a couple photos of Guinea. And that is the room. Thank you.
Oh, I just closed everything. Hold on just a second. I think so.
Okay.
File that's titled the poliovirus 2. Is that poliovirus 2?
Yeah.
Okay, guys. It's time for my epic presentation on the woke mind virus. This is a very unfortunate virus that infects many, many people and makes them woke and makes them pronouns. I'm very funny. Anyways, I'm just kidding. It's about polio. Yeah, I'm very funny. Please laugh. Haha. Funny, funny, funny. Anyways. Okay, so it's actually poliovirus. Poliomyelitis is the thing it causes. But poliovirus is part of enterovirus. It's a type of enterovirus with a subgroup in the, I'm not really, Picornaviridae family. Hard to pronounce. It's a single-stranded RNA virus. And our 3 different strains, there's only one that's still alive, and that one's just in like developing countries right now. The other 2 were killed off in 1999 and 2012 respectively, although they might still be in labs somewhere. I don't know. Maybe. It's icosahedral shaped, so it has 20 sides like this. It looks like a poorly rendered sphere in a PS2 game, and then for size, it is this one right here, the third to last one, so it's really, really tiny, really small. And there's a virus right there. The virus is huge. So the main thing about it that everyone knows is that it's neurotropic. Well, I mean, people don't really know that word, but they do know that it causes paralysis because of the fact that it's neurotropic. It kills motor neuron cells, which causes the notorious paralysis symptoms, like I said. And it's very lytic, which just means that it kills the motor neurons. I already said that. And it inhibits DNA transcription, translation, which sort of slows down the interferon response. But the big thing is the fact that it's a foggy species. It mutates a lot. It doesn't really restrict itself from having errors whenever it replicates. So that's the main problem with it. And that's probably why it spread so much, because people couldn't really adapt to it as well. Now, the main way people get it is the fecal-oral route, which this is just one way that the fecal-oral route can happen, which is that someone poops, then they don't wash their hands correctly, then they touch food, and then someone else eats that food. But it can happen in other ways. Currently, in developing countries, the main reason why it's still there is because a lot of the water there is contaminated with fecal matter. There's also the oral route, which is, mouth to mouth, like kissing, sharing food, stuff like that. Since it spreads through droplets, aerosols, stuff like that, and of course feces. So the main people at risk are going to be children, people under 5 years of age, unvaccinated people, obviously. People who have recently visited an endemic country, or those who live in an endemic country, or immunodeficient people, of course. There's a few different kinds of polio. There's the abortive one, which is where you just get flu-like symptoms, fever, headaches, aches, sore throat, loss of appetite, nausea, vomiting, average stuff. And there's non-paralytic polio, which has all the symptoms of abortive polio, plus some muscle weakness, stiffness, pain. And a second phase can happen within a few days after, which causes stiffness in the neck and spine, and decreased reflexes, and muscle weakness, and all that stuff. And this is when it's at its worst, is paralytic polio is the most severe form, and while it is rare, it does happen. And it's gonna have all the symptoms of non-paralytic polio, but it slowly gets even worse until possibly you get paralysis in limbs and such. And the reason why it kills is because it can paralyze the lungs and airway muscles, which keeps you from breathing and kills you. There's also post-polio symptom, which occurs decades after you get polio, or can happen. Includes muscle joint weakness, pain, fatigue, muscle wasting, breathing, swallowing problems, sleep apnea, and sensitivity to temperatures. And also 2 to 3% of polio cases can also cause meningitis. The most mild forms of polio can easily be mistaken for the flu, of course, like I said, flu-like symptoms. But some neurological diseases can also be mistaken for polio and vice versa. For example, I'm not sure how to pronounce that, Guillain-Barré syndrome, spinal tumors, and acute flaccid myelitis, which, to be fair, acute flaccid myelitis can be caused by polio, but not always. And some people may not even realize that they have it at all because it just happens and you have no symptoms and then it goes away. So the main diagnostic method is going to be isolation. This is where they do a PCR basically on a stool sample. And this can also help distinguish wild polio from vaccine polio in a process called intratypic differentiation. Like I said, it's the most common method and can also be used to find the geological region of the wild polio if needed. There's also serology testing, which is trying to find the antibody. The specimen can be collected from the patient, and it can support a polio diagnosis, but it's not really absolute. And it's mainly only really used if the patient is suspected to be unvaccinated. And then the most rare form of diagnostic testing is going to be cerebrospinal fluid analysis, which can be looked at for any abnormalities. But a lot of these abnormalities can be explained with other stuff, so it doesn't really narrow things down. It just kind of supports it as well, supports the diagnosis. There isn't really any treatment. If you get it, it's over. Kind of. Not necessarily. But you can get physical therapy, or you can get occupational therapy if it's so bad that you literally like can't do everyday tasks. And then there's a couple of vaccines that were made against it in the 50s. There's the oral one, which is an live attenuated vaccine. It's taken orally since polio is also contracted through the mouth and in the intestines, so that helps the intestines make the antibodies first. Problem with that though, since it's live attenuated, it can easily revert back to the normal virus, and it can happen in like I think around 1 in 2 million people who get it. It can revert to the normal virus and start replicating and become regular polio. But the main one that's used is the inactivated polio virus, which encourages the body to make antibodies in the bloodstream. And I mean, yeah, it doesn't really go into the intestines, but it still works the same and it's generally safer. So it's just the better option, just hurts a little bit more. And then finally, we got the iron lung. Of course, I gotta talk about this. This was used to treat polio for when people had their airways and lungs paralyzed. Basically, literally pushes air in and out of you, doing the work that your lung is doing by literally pushing your lungs. It was first used in 1928 and then ceased use in the 1950s, whenever the vaccine was made and there was no need for the use of it. However, there was one person that was still using the iron lung up until this year. He was still in the iron lung. His name was Paul Alexander. He was in the iron lung since he was 6 and died at 78. So he was in the iron lung for literally 72 years. But somehow he still lived a very successful life. And I believe he was like a lawyer and an author, and he wrote books. I think he was popular on TikTok at some point. He passed away.
Very neat, you know.
That's a funnier joke than the wolf virus joke. I thought the wolf virus was a pretty funny joke. Very funny, haha. By the way, I don't unironically use the term wolf. It's a joke. Haha. Anyways. Like I said, he died March 11th, 2024, and he had a pretty good life. Which you wouldn't think that someone who was in an iron lung would, but he seemed very happy and very fulfilled. You can watch lots of videos on it on YouTube if you're curious. And I think that's pretty much it. And to quote the great Franklin D. Roosevelt, he said, "We have nothing to fear but big scary monsters." Ah, that has nothing to do with polio. I just added it there. Was I funny?
Yes, you were.
Yay. Easy.
All this blood, I was like, for
Huh?
FDR, man, in the butt of our jokes today.
Did I do good?
You did amazing.
You did great,
That's
Grace.
I did?
Yeah, yeah. You really did, I'm being honest. Okay, next we have. You're fine, I know, yeah.
And then
I had struggling.
Well, that is so cute.
Yeah.
Alright, guys, bear with me. There's a couple words in here that I'm not 110%
sure how to pronounce, so we're just gonna roll with it. Today I am gonna be going over rabies, which is, just essentially a slow progressive zoonotic disease, which results in, fetal encephalitis, swelling of the brain. So the etological agent of it is in the Rhabdoviridae family, and it is a lysovirus, which is a RNA virus. It does have this really unique bullet-like shape that is round on one end and flat on the other. It is also helical encapsulated, and it has spikes that protrude through the outside of it, which kind of, helps it adhere. For the variance factors, I had a really hard time finding any information for this, but what I did get is the fact that the gene itself does encode 5 different proteins: the nucleoprotein, the phosphoprotein, matrix protein, glycoprotein, and large protein. The 2 important ones out of the 5 would be the nucleoprotein. That's basically the protector of the organism. It helps it survive. And then you have the glycoprotein. That is what allows it. It's very essential for the pathogenicity of the virus and the invasiveness, which allows it to, take over. Any mutations that occur to the amino acid sequence for the virus can either be very devastating, or it could help it tremendously depending on how it goes. What they do know is the fact that it can inhabit the CD8 plus T cells and the inflammation response, which the T cells are usually what kills viruses, which is why it's so dangerous. They don't, however, know how it has the ability to do that. So the steps of infection, obviously, is by getting the saliva into your body. That's mainly from, animal bites, but it can, in rare cases, be inhaled or be inoculated through the membranes of the eye. It remains at the infection site for long periods of time. And that's where it multiplies and just gets bigger. And then it gradually starts to spread out until it injures your brain and nervous system. From there, it infects your eyes, heart, skin, oral cavities, and then it completes its cycle once it has penetrated the salivary glands and can be presented in your saliva. So modes of transport, the most commonly one are dog bites. They are from, wild animals. So animals get them from, bats is one that's commonly mentioned. I know when I was younger, my sister and I got chased by a rabid skunk, and that was a fun experience. So they usually get infected from that, and then they come in and they infect a human. It's not very common in the US, but there are, cases seen between 35,000 and 50,000 annually. Children, travelers, animal handlers, and pet owners are obviously the ones higher at risk, and anybody who has to deal with animals has the chance of getting it. For patients who do get the rabies virus, there are 2 different categories that they can fall under. You have the serious, which is the one that presents the aggressive behavior, and then you have what they call dumb, which is just a state of confusion for patients. You can have seizures, twitching. For the furious category, I would say the most prominent symptom would be the hydrophobia. That's where they literally develop a fear of water, so they don't drink it, they don't do anything with it because the pain that they feel in their throat is so excruciating that it scares them away. For the furious patients, they are fully coherent of what's going on. So, whereas the dumb patients are confused and not really comprehensive of what's happening. So some different, some different diagnoses. So if a patient comes in and they haven't like reported that they were attacked or symptoms aren't present, it's very easy to misdiagnose rabies. Most of the underlying symptoms like the anxiety and depression that is seen is easily overlooked. Some of the bigger symptoms like the muscle spasms is usually related to tetanus, and the encephalitis can mimic a whole bunch of different viral infections. Usually the diagnosis isn't made until the autopsy is done after the patient has died. Sorry. So the diagnostic methods, there are a ton of different ones. For the direct fluorescent antibody testing and the direct rapid testing, the most useful part is brain tissue. That's where you're going to find the most concentrated antigen for the virus. The direct fluorescent antibody, it is very important. The fluorescent part of it is the anti-rabies antibody that is injected, and it binds to the antigen. And then when you wash away everything else, the antigen, it stays where it's at, and it glows that really bright kind of apple green color to show you that the rabies antigen is there. Another one is the immunohistochemistry test. That is a newer diagnostic test. And it uses the rabies genetic material to diagnose. So what it does is you use the, it's like a cross section of the spinal cord and the cerebrum for patients who haven't succumbed to the disease yet. You can do like saliva and a skin biopsy from the nape of the neck. And what you do is it's like a single tube reaction that you use the viral genetic material, and then it amplifies into several different copies, and it can be detected by fluorescent probes. Let's see. Oop. Look at me hitting buttons. It like scrolled off the page for some reason. So that's the picture of it at the bottom. That's the picture of that one with the tubes. That particular test, it targets different regions of the virus. So like the leader region in the virus, and then it also targets the nucleoprotein gene that we talked about earlier. For treatments and prevention, the best way to prevent it, you know, is to make sure you vaccinate your animals, or for individuals who work closely with animals, you can do the vaccinations also. Eliminating the amount of strays that are in the surrounding area, and then doing a strict quarantine of any potential cases. For treatment, it is essential that anybody who does get infected seeks treatment immediately. You have to start those vaccines like before symptoms are shown. So usually what they do is they will infuse the infection site with the human rabies immune globulin. And then you undergo a rigorous vaccination cycle, which is usually injected either through intramuscularly or intradermally on the first, third, seventh, and 14th days. And then sometimes if you put patients in drug-induced coma or with a ventilator, it can save their life too. I think that is all I
Well, no, she's ready. I don't know how much I love it, but I find it so fast.
Wish I chose rabies, that would've been fun.
It's a really good
Hold
one.
on before you switch over.
Uh-huh, right. Uh-huh,
That's the only way. So for animals,
Yeah.
they don't have an ability testing for rabies.
you should be able to click or spacebar or arrow. Okay. I'm gonna have to click on the screen first, sorry. Oh, okay. Yeah, I mean,
That's what it's
Yeah.
fascinating.
if you have like a dog that like, let's say your dog is out and some kids like start throwing rocks at it and dog bites one of those kids. If you have a pet, they'll keep them in the vet for 10 days. If they show any signs of rabies whatsoever, aggression, foaming at the mouth, anything like that, they'll just put it down. They just don't even test it. So that's how that goes. It's because it is so serious of the disease. But yeah, so with animals, they don't really do a lot of testing for it. They're just gonna assume that it has rabies and they'll test for it if there's a human involved in it to cross check to be sure that person needs to receive the treatment. The treatments are actually can be very intensive. Or you don't just get a vaccine. You get vaccines in your abdomen because that's where your high dislocation of your white blood cells are gonna be to like get involved in that. And the bigger infusion of immune globulin as well. Right, to give you that passive protective immunity is a lot. So rabies is it's no joke. We think of it as being like, we don't think about our pets are vaccinated, but in other countries, they have wild dogs running around that cause rabies. So it's a bigger deal. It's more connected. You go into like rural parts of Mexico that aren't money. So even if you spend a lot of city, they'll have a lot of wild dogs and rabies.
Yeah.
So you just have to be aware in this area. Stay away from wild dogs, but like our wild dogs probably don't have rabies. Theirs probably do. So it just puts it in perspective, how kind of dangerous we are here in the United States for diseases like that. Not just rabies, but other ones that you mentioned, that trachoma, with the eye infection that we just don't think about here in the US. Chlamydia in the eye doesn't really affect it. So that would be so weird, but you know, sorry.
No, you're good. No, you're fine. My name's Hannah. I'm in Professor Yost High Group class, so I don't know any of you, but thank you
Okay.
for just letting me. Yeah, I'm from her high group class Monday, Wednesday. So, I'm going to be talking over the African horse sickness. Get into it. Okay, so just a few facts. It is prevalent in North and West Africa due to the name. It's infectious but not contagious. Its genus is Orbivorous. It does have a double-stranded RNA. And it does have a 3-layer capsid, like the cell envelopes. It'll be the inner capsid, the core, and then the outer capsid as well. Some virulence factors, basically just explaining how it got there. The main point is that the NS4, it surpasses innate immunity, and it just basically promotes replication. It will that NS4 will show up in the heart, lung, and spleen, so it's something to look out for there. Mode of transmission, just basically saying how we did get there. It is mainly by insect bites, especially those biting midges. Yeah, midges, those are like the number one insect that infects the horses. Also ticks, contaminated needles, if they're dirty, bloody, any needles, and then they are most often found in farms and just greenery and where horses like to hang out. Some risk factors, obviously, is climate. So where you keep your horse, some environmental warnings, if it's warm, wet weather, or if there's a drought, that, droughts especially because they, that's where those midges like to, hang out, and so, if you live somewhere that's definitely, has a lot of droughts, you're gonna attract midges. And then seasonal disease, it's endemic to obviously Saharan Africa. Again, it's in the name, so those are some risk factors. Signs, symptoms, and diseases. Some signs and symptoms, obviously redness of eyes, colic, depression, their face swells, it's really sad, just in decline, and then a disease it's mistaken for is going to be that blue tongue virus as well. Diagnostic methods, there was a huge list, surprisingly, and so I kind of just condensed it. It showed up with the ELISA, PCR, some virus isolation, and then also that RT reverse transcription. Again, I condensed it, there was a longer list. Okay, Fix-It-Felix method. There is no known cure. It's really sad. You just kind of watch your horse decline. But you can, there's some vaccines. But normally the best thing to do for your horse is just to take care of it. Just give it rest, a good environment, safety, attention. But there's nothing you can really do, sadly. But and that's it. Thanks, guys.
No, that
It
was
was to be funny.
Oh, okay.
That wasn't the official memo.
No, that was just. No, where did she?
All right, Mackenzie. You ready? This one, right?
Yeah.
That you can click, which is like the left click, or arrows or space bar should work.
Okay, guys, I'm Mackenzie. I'm also in the hybrid, so I don't know any of you either. I think I have a few before. So we're going over the canine parvovirus. Pretty sad, but treatable, so we're good. All right, what exactly is parvo? It comes from the carnivore protoparvirus 1 strand, but there's a ton of mutated different strands of it. But it does pretty much stay within dogs, coyotes, wolves, even foxes can get it though, which is a little weird. So it is non-enveloped, and it has a spherical capsid. Virus, which sucks. And so when it is ingested, it gets into the gastro tract, and that's where it mostly hangs out, and it replicates in those rapidly dividing cells. So it spreads after hanging out for a little bit. And how do we get there? Whale, teeth, of course. So contaminated surfaces, and it is resistant to humidity, heat, and cold. The problem with this is if, like, say 6 months ago, an infected dog came by through your yard, pottied, and then kind of went on its way, even after like 6-7 months, and you know it's rained, it snowed, it's Oklahoma, so probably some natives, you know, all the things, it's still there. It's still probable to give your dog. So if you're cute little puppy goes out, goes potty, steps in it, comes in later, it's licking its paw. It's there. Ignore my shakiness, guys, sorry. Okay, so I am not a health speaker. Alright, so who or what's at risk? Dog. Kind of already went over that. Puppies are the ones that are most at risk. Unfortunately, any less than 6 months are definitely most at risk. Any dog can get it, but seniors and puppies are the most prone to it. Or if your dog is unvaccinated or poorly vaxxed, which is crazy because they're pretty cheap at the vet. And you can go to Atwood's, Tractor Supply, PetSmart, they all have them there. So there's not really an excuse for that one. Winter worry. So it doesn't even show up till 3 to 7 days later. You won't even know. And which makes for a lot of cleaning. So the first signs are usually appetite loss, and then, just sleepy. And then you have to be really quick to catch the fever because even though it's on this list, it spikes and then immediately their temperatures just start dropping rapidly. And that happens most commonly in smaller puppies. And they present more extremely because it also affects their heart muscles due to it being a rapidly dividing cell thing. Yeah. So what it mimics, when you take your dog to the vet or it first shows signs, they're immediately going to think parvo anyways, just because it's such a serious disease. But it does also kind of look like canine distemper. I can't say that one, but gastroenteritis and other, just like tummy aches and stuff like that. Like if they eat something bad, since they're vomiting, nausea, all that stuff, just laying around, it could just be a tummy bug, but they're gonna test for parvo first. So how do they test for it? They will take a blood sample, and if they don't have the ability to do it in vet, they send it off. And what they're testing is it's basically the plasma of it. But yeah, so in the vet they can do what is a snap test, and that's where they get a fecal sample and it pops up this little dot on the stick, and yeah, it's positive, and that's not good. And then you have your PCR. So different ways, and they do all of them. They don't just stop at one. So how do we fix it? Well, there's not really a cure for it, unfortunately. So it's just symptomatic care. If they have a fever, they'll push fluids. In small puppies and in older seniors, they really check their glycemic levels because those are known to drop tremendously. And they'll monitor intake versus output. So yeah. The best thing for it though is vaccination, and they will vaccinate your dog while it's in the vet. And that's it. And Ginger Maverick sleeping.
If you ladies want to stay, you're welcome to, but you don't have to. It's not required. We'll try to keep things brief today on my end. All right, so last time we talked about our skin and eye diseases, and when we did that, we got into this concept of immune privilege, and immune privilege being our down regulation of the immune response in the eye, specifically to prevent damage to the eye tissues and preserve eyesight, right? And so we have down regulation of our lymphocyte response and as well as mediation of our innate response as well. But looking at it that way, fine. The CNS specifically is going to have a similar aspect for downregulating our immune response. The overall nervous system will have a similar effect, but particularly in the brain, where we're talking about the blood-brain barrier. This is a literal, actual physical aspect of barrier between the brain and the bloodstream. We have the endothelial cells of the bloodstream being, thicker and moderating things that can pass through from the bloodstream into the brain. They're not as able to do that in the brain as they are in other tissues of the body. Of course, there are some drugs that are still, able to cross that barrier, and there are also some microbes that have found ways to migrate across that barrier as well. But overall, generally speaking, it protects us from a lot of infectious diseases and prevents our inflammatory immune response from causing damage in our central nervous system. In our central nervous system, we'll have our own cells that are going to be tasked with providing our immune response in those areas, like certain we'll say innate cells, that macrophage-like cells that will work in the brain only, and help prevent that damage that we might have in our nervous system. I'm not going to go too much over the nervous system. You guys learn mostly about that in if you're taking anatomy in those anatomy classes, but these are going to be nerves that are involved in senses and signaling and memory, as well as nerves that can transition between the two. So, and give signals to muscles and movement and all that. We have the coating of our brain, which is called the meninges. This is made up of 3 essential versions. We have the dura mater, the arachnoid mater, and the pia mater. So these guys are all going to be involved in providing that protective layer over our central nervous system, our brain and our spinal cord in particular. The skull also provides some protection as well for shock, and the fluid, the CSF itself, also helps with that. The CSF should be completely sterile. In fact, all of your entire nervous system should be completely sterile. There is absolutely no microbiome in these areas. For obvious reasons, we wouldn't want to risk overgrowth of one or another and then have to deal with an immune response to that. So we can't afford to even have microbial antagonism in place with taking up our stadium seats. All of our stadium seats in our nervous system are completely open, but the goal is to keep them that way. We don't want invaders taking them up either. And right, we talked about this blood-brain barrier. So our immune privilege, again, preventing damage to the central nervous system. If you damage a nerve that's part of your central nervous system, you cannot heal that nerve. It's damaged permanently. There's no way at this time for your body to naturally heal from that. In fact, science is having a hard time even forcing that to happen. So once damage is done to your central nervous system, it is done. So that is why we have to be so careful about having our immune response kick into gear. You don't want too much activation of a complement, poking holes in our own cells on accident, or anything like that could lead to paralysis and loss in senses. So we have microglial cells that are our phagocytes, brain macrophages, like I was saying, these are really our phagocytic cells as well in the central nervous system. And both of these are actually going to have way less reactivity than what we would see in normal macrophages outside of the CNS. So it's hard to protect these areas, and they have to have their specialized cells to do so. That's why that blood-brain barrier is so important, keeping infectious agents out so that we never have to even deal with them. Okay, so it should be lacking any biota. We're going to kick right into some of our diseases. There are nervous system aspects of COVID that we can think about. COVID itself, we're considering it a cardiovascular disease, by the way. So it'll be in the next chapter overall, but you can have neurological symptoms associated with it. We know altered smell and taste. That's a nervous system issue. So not being able to taste things or smell things, headache and confusion, and certain movement problems associated with COVID also fall into the neurological category. Meningitis, this is a syndrome, an anatomical syndrome. That means we are classifying it based on our actual signs and symptoms, not on what causes it, right? So if you talk about you have the flu, what causes the flu? The flu virus. Like that's it. That's all that causes it. With meningitis, what can cause meningitis? Well, you have different kinds of bacteria, different fungi, and different viruses, all of which that can contribute to the development of meningitis. Itis means inflammation always. Anytime you see that at the end of something, inflammation immediately. To actual active inflammation of the meninges, the coating, all those mater layers that we have protecting our central nervous system. So here we will have things like photophobia, which is light sensitivity. We'll have fever, headache, pain and stiffness of the neck, all of these associated with meningitis. And the way we're going to be looking at our patients, they come in with any of those symptoms, especially if they have a rash associated with that, which may or may not be present with meningitis. We're going to be doing a lumbar puncture immediately. If you come in with a headache and neck pain and photophobia, we're going to do a lumbar puncture. We're not even going to wait. And we're going to look at that in gram stain and see if there's any microbes in there. In the gram stain, you'll be able to tell if there are fungi present as well, not just bacteria. We know gram stains, we use them to differentiate between gram positive and gram negative bacteria, but you're also potentially going to be staining any fungi that could be there. So already with the gram stain, you can rule out our big categories. Is it a bacteria? Is it a fungus? Or is it a virus? If somebody's presenting with meningitis and you're very sure that it is meningitis and you don't see bacteria or fungus, it's probably a virus. So you won't see those on our typical microscopes for a gram stain. If it stains gram positive or negative for bacteria, then we can start determining what the actual cause is most likely to be based on the frequency of certain diseases associated with it. So that's our number one line of diagnostics for meningitis is our lumbar puncture and our gram stain. We're not going to wait though. You come in with meningitis, we're not going to care if it's bacterial, fungal, or viral or anything. We're going to start pumping you full of a broad spectrum of antibiotics right off the bat to try to start and stem the progress of that disease before you die, because we might do that before you get your treatment, your results back from your testing. So these are a summary of our symptoms we just talked about. Certain microbes that are involved in causing meningitis will have their own specific virulence factors, whether that's capsules or toxins they produce or antibiotic resistance or whatever it is. They each have their own virulence factors, but the symptoms and the signs will be, generally speaking, the same no matter what the cause. Okay, so getting into the bacterial meningitis, this is Neisseria meningitidis. Neisseria meningitidis is very closely, same genus as Neisseria gonorrhea. So both of those would be gram-negative diplococci, all right? So with meningitis, so this is our meningococcus. You've probably heard of it called that before. The meningococcus is a gram-negative diplococcus. That feature of it is extremely important for these guys, okay? This is where if I do your lumbar puncture, I go look at your gram stain and you have gram-negative diplococci bacteria, you have Neisseria meningitidis as your cause of your meningitis. It is the most serious form that you can get of meningitis overall by far. About 14% of cases of meningitis will be caused by this. So it's actually pretty common. Most meningitis in general is going to be caused by viral causes that will you'll recover on your own. But this one is the most serious one by far, and it's bacterial. So you'll get a rash associated with this, very commonly a petechial rash. So we have bursting of blood vessels under the skin. And the bursting of those blood vessels can eventually lead to necrosis of tissue in the areas as well. So this is a baby that had the meningococcal meningitis that progressed into meningococcemia. That means that the bacteria spread throughout the entire body and became systemic, not just in the nervous system. So we have necrosis of tissue associated with the destruction of cells and vasculature for the spread of this disease. It's incredibly deadly disease, high mortality rate. So that's why we give antibiotics right away. It's because if we suspect it's Neisseria meningitidis, our meningococcus, that's what we want to stop immediately. That's our number one concern. Second to that is Streptococcus pneumoniae. It's the gram-positive bacteria. It's a coccus, but it's a strepto. So we have chains usually. It'll usually be in pairs or in chains. And we call this one the pneumococcus. You guys probably heard this one as well. So pneumococcal meningitis is a community-associated meningitis. So is the other one, but this one is more common. We're more likely to get this than the meningococcus one, okay? So this one's the gram-positive, the other one's the gram-negative. The gram-negative is more serious. Gram-positive, it's still really bad though, high mortality rates. Just no rash associated with this one, okay? Most common cause of communally acquired meningitis by far. So being around other people. When I say community acquired, I'm talking about getting it from the people that you're around day to day. Generally speaking, there's either two things you can get these infectious diseases from: community acquired or hospital associated. So that's what the differentiation is going to be in a lot of these diseases that we talk about through the whole unit. So this is community associated, so being around other people, just out day-to-day stuff. Usually going to have a worse association with it if you have some sort of downregulation of your immune response. So again, we'll do our gram stain. If it's gram positive cocci, then it's probably this one, and we'll treat it accordingly. If it is gram negative, it's probably the worst one. They both of them have capsules that can be aiding in their virulence as well. There are vaccines that are available for the pneumococcus. So if you are going into like college and staying in dorms and stuff, they'll actually require that you have vaccines for meningococcal versus pneumococcal. The older that you get, they're going to push more pneumococcal. So yeah, there's both of them have vaccines. But all right, next one is Haemophilus influenzae, another bacterium that causes meningitis. It has another vaccine called the Hib. So HI, Haemophilus influenzae, serotype B. We sometimes see A associated with it, but it's mostly B. So Hib is the more common one, has a vaccine for that one. Listeria monocytogenes is a gram-positive rod that's modal that is associated with food transmission. This is what you'll get. You're supposed to microwave if you're pregnant. You're supposed to microwave your lunch meats and stuff to warm them up and kill any bacteria that could be on them. Hot dogs, not eating them raw, things like that. These guys kind of are pretty hardy, and they'll grow on like your colder meats and things like that. You eat them, it gets into your body through like your gut, and you can have gut-associated symptoms. It's not a big deal for people who have a healthy immune system. Otherwise, it can get into your actual nervous system. It'll infect your insides of your cells. It'll get into your cells and then migrate through the actual cells across your blood-brain barrier and get into your brain and cause meningitis. It's a lot more serious for pregnant women because it can cross the placental barrier as well and cause issues with the babies due to miscarriage and stillbirth and stuff. That would be listeriosis. So any infection with listeria is listeriosis. And then we have listeria associated meningitis that is a separate like version of it. Pretty high mortality rate, but I've already told you this is less serious than what we would see with the meningococcus or the pneumococcus. So still pretty serious. 20% is pretty high, guys. This is what we're talking about with like anthrax. So this is our low level of anthrax, of course, but still pretty serious stuff. So you do want to be sure you're cooking your food. All right, now we're moving into the fungal meningitis. This is Cryptococcus neoformans. So Cryptococcus is a fungus, usually associated with bird droppings. We see this a lot more in AIDS patients. So it's a more chronic development, whereas Coccidioides immitis. So Coccidioides immitis is, oh my gosh, coccidioidomycosis is the disease that it causes. And the disease that it also has a, I guess colloquial name of valley fever. It's a fungal infection as well. It can cause meningitis, but can cause other symptoms as well. It's not always meningitis related. A person like California, Arizona, southern, actual true Southwest area. So it's mostly in Arizona that we'll see this popping up. Fungal. Viral meningitis, like I said, it's usually you're going to recover over it on your own. 90% of them are caused by enteroviruses. Enterovirus is anything that gets into the gut. Like if you guys weren't paying attention to Grace's presentation, polio is an enterovirus. You get it by ingesting it, and then it moves into the nervous system. So that's an enterovirus. So other ones that are like that might be things like polio and how they're transmitted. We also have things like herpes that can actually cause meningitis. It isn't necessarily an enterovirus, but can still lead to the development of hepatitis, or of viral meningitis. So herpes virus and other HSV2, herpes simplex virus 2, which is our genital herpes, as well as a lot of other herpes viruses, including human herpes virus 3, which is also known as varicella zoster, which is also the cause of chickenpox and shingles. It can all cause meningitis depending on how it progresses. So yeah, usually you're going to be getting over this on your own. Mortality rate is less than 1% with viral meningitis. Nobody really gives a crap about it if you have it. So you have viral meningitis, we'll treat your inflammation associated with it, but you'll get over it, basically, if you have an appropriate immune response. It's more severe, of course, if you are an infant and you don't have your own immune system yet. And so you can get meningitis caused by other bacteria that wouldn't cause it in adults that have an immune system. So this can be caused by Streptococcus agalactiae and E. coli, and we already talked about Listeria. But the Strep agalactiae, that one is one that they will be testing for later in pregnancy to see if you have the strep bacteria. And if you do, they will give you antibiotics before you give birth to protect from the development of meningitis. Yes.
I have something on that. So my nephew's partner at the time had strep when she gave birth.
Mhm.
They did not test her. Gave birth to my nephew, which caused PANDAS.
Mhm.
Like, it's severe, like schizo, Tourette's, all these other things. And they've thought about draining his blood to clean it, to maybe try
Give
to
transfusions to give a better, wow,
kind of calm it down
because of the neurological effects of it.
Yeah.
Wow, that is
he had this
crazy.
so severe.
Yeah, that is crazy.
pain.
So yeah, we don't always think about the effects that we think of meningitis. Okay, you get sick with the thing and then you get meningitis. We forget about the long-term effects that having an illness can have on your entire nervous system, including your mental functions. So that can include things like you would never expect a mental illness to be associated with some of these things, but presentation of those symptoms can be associated with anything, you know, causing an issue with your brain function, including infections. But yes, 20 to 30% of females just have this normally in their genital tract, for example. So we can test this in your vaginal canal. You're supposed to be testing for it later on in like the 3rd trimester to see if your patients have this, and you can give penicillin-based antibiotics to treat it. I'm not going to go over all the details of that. All right, meningoencephalitis, we have encephalitis means inflammation of encephaly, which is the brain. So we have inflammation of the brain and the meninges in meningoencephalitis, okay? The only one that you need to worry about for meningoencephalitis, well, there's 2 causes, but they're both protozoa, okay? Both amoebas. This is the brain-eating amoeba disease. So when I was telling you guys about your contacts and washing your hands and acanthamoeba, well, here it is again, acanthamoeba. So if it gets into your nervous system, it can cause the brain-eating amoeba disease, which is AKA, for our purposes, meningoencephalitis. Okay, so that's what this is talking about. Both of these guys are found in fresh water, warm, fresh bodies of water. It doesn't need to be actually fresh, but you know what I mean. It's not salinated, right? Might be nasty. Probably is. Go swimming in the lake over your 4th of July weekend. You get water up into your sinuses, and it can get into your brain through your blood-brain barrier that way, or through contacts and the abrasions that they can cause as well. So they will enter into the brain and cause disease. Naegleria fowleri is going to cause the faster, worse version of this, but they're both hella bad, okay? Both of these guys have close to the same mortality rate. It's just this one will present faster. And this one's the one associated with like lakes and stuff and getting in the olfactory way, whereas acanthamoeba is going to be associated with like contact use and things like that. Naegleria causes the form called primary amoebic meningoencephalitis, or PAM. 97% mortality rate, very fast progression, almost no treatment can be offered other than palliative care. And you will die within usually a couple weeks. Don't use non-sterile neti pot water. You're going to use a neti pot, which I do highly recommend, and most ENTs will recommend that you do. Use like actual bottled distilled water. Heat it up in your microwave or a kettle that has been cleaned, boil it and let it cool down. And then you can use your neti pot. But if you're going to use neti pot, don't use just straight up tap water. Okay. If you use a neti pot, you're supposed to be adding salt to it, just to be clear, like salt, usually bicarb solution as well, to make it like isotonic, for that word. So talk to your ENTs if you're going to do that. It helps a ton with sinus infections. Okay, acanthamoeba, this one causes GAM, or granulomatous amoebic meningoencephalitis. So granulomas are whenever your white blood cells will wall off infection, basically to protect from spreading. So that's a granuloma. So this has presentation with granuloma. This is a little bit more slow going, but it's still 97% mortality rate, ultimately. All right, encephalitis. Typically, when we talk about encephalitis by itself, we're talking about acute encephalitis. So these are brain infections or brain-related infections. Almost all of the ones that we're going to talk about today are going to be associated with like mosquito-borne viruses. Okay, so this might be like, wow, acute encephalitis, mosquito-borne viruses. This sounds crazy. West Nile virus. Okay, you guys know West Nile virus. All over the place. Most people will have had it at some point here in Oklahoma, but not ever even had symptoms. You would never even know if you had it most of the time. Some people will get headaches or true encephalitis associated with it, but it's pretty uncommon to see the development of that. So these things can be very mild. They may have symptoms of meningitis as well. We'll treat this with acyclovir, which we'll remember we learned about with herpes viruses. Remember valcyclovir or Valtrex? Well, acyclovir is pretty a wide use antiviral. These viruses that are transmitted by mosquitoes are called arboviruses. So I always think of arbo as being like a tree, like Arbor Day, you know? So arbo being like a tree that's up in the air like mosquitoes are, okay? So that's how I remember these are mosquito-borne for the most part, but these could be any bug bites. It could be ticks or anything like that. So usually mosquitoes though, and so they're going to, we're gonna see these diseases coming out during the seasons where we have mosquitoes. Transmitted through the blood, we have West Nile, St. Louis. You'll see the distribution in the green in the maps and where the reservoirs are. Normally maybe found in birds, and then the mosquitoes will transmit it from the birds to us. Lacrosse virus, Powassan virus, and some of these are far more serious than other ones. Jamestown, Eastern equine, the case fatality rate in those who have symptoms from Eastern equine, by the way, if you're a human, is 70% fatality rate. That is impressive and terrifying. All right, next we're moving on to herpes. Herpes virus can cause encephalitis. We already said how it could cause meningitis, but it also can be associated with encephalitis. Can be one or both. Encephalitis is usually HSV1 most commonly, whereas the meningitis is usually HSV2, but it doesn't really matter too much on that, okay? But herpes can cross into the actual brain and lead to that. We already know herpes hides out in your nerves. Not surprising it could move into the brain. Now we're going to move into the subacute encephalitis. These are guys that will have like less severe symptoms, less striking, but the disease is going to last for much longer. So we have toxoplasma, measles, and prions. Toxoplasma, primarily we're talking about Toxoplasma gondii. Toxoplasmosis is when you have this protozoa, it's protozoa, and it will be found in cats that are a reservoir. And they poop in the litter box, and you're pregnant, and you pick up the litter, and you get the protozoa into your body. Most people will get exposed to toxoplasma at some point in their life if they come into contact with cats. Very, very common. You may or may not have symptoms from infection. Turns out Toxoplasma gondii and toxoplasmosis. Let's see where it starts together. It is cats. Toxoplasmosis and Toxoplasma gondii being the reservoir in cats. When mice get this, it controls how their brains function and makes them more likely to take risk-taking behavior. So less likely to be deterred by a cat being in the area, making them more likely to be eaten by the cat, and therefore keeping the life cycle of toxoplasma in rotation, right? So mouse is getting eaten by the cat, cat gets infected, toxoplasma wins, right? Has the same effect on humans. You're more likely to take risk-taking behaviors. What are the odds that maybe one of your presidents or you or somebody else in a managerial or even higher up position anywhere that you've known may have been infected with toxoplasma, and toxoplasma is actually running the situation and not the person? Interesting to think about. It's a conspiracy theory thing. So, I mean, I bring it up because it does actually cause risk-taking behaviors to increase in people who are infected. And it's not a symptom that you would think of as being from an infectious disease, right? It's a personality thing more than anything. You usually clear this if you have a healthy immune system. It won't hang around. You'll probably never know if you had it. People who are pregnant, this can transmit to the fetus and be deadly or cause permanent birth defects in a fetus. So that's why we say if you're pregnant, don't handle the cat litter. Has otherwise like cold flu-like symptoms associated with this. We could have stillbirth, liver and spleen enlargement, liver failure, hydrocephalus, convulsions, and damage to the retina and permanent blindness of the fetus whenever it's born, if it survives that. So this is in congenital infections. So the baby in the womb. 33% chance that if you get infected with toxoplasma during your pregnancy, that you're going to spread it to your child. A 1 in 3 chance. Don't risk it. Don't handle the litter. Get somebody else to do it. If you have a cat, pay for a service. You don't want to be doing that. You don't want to take a 1 in 3 chance that you're going to lose your baby. Measles can also cause encephalitis. I'm just going to leave it at that, guys. Prions, we've already introduced Kuru, but this includes mad cow disease, Creutzfeldt-Jakob disease, which is mad cow in people. That can be acquired from eating mad cow meat, or it can be just like random. You can have like a predisposition for having prion diseases based on certain genetic factors, but typically speaking, this is going to be transmitted by misfolded proteins being introduced into your body, essentially. And fatal familial insomnia, literally your proteins will get misfolded and won't be able to work the way they're supposed to. These are proteins that are associated with helping you fall asleep, and you literally cannot fall asleep anymore eventually. And you die from not being able to fall asleep and allow your body to repair itself during your sleep process. That's what you die from. Forget it. I'm not. Yeah, it's terrifying, but it'll run in your family, so you would know if that was coming. It's not going to be like a surprise. All misfolded proteins, there is no genetic factor for the prion itself, but you can have genetics that can predispose your protein to being misfolded, if that makes sense. Zika virus, this is another one that's transmitted by mosquitoes. It leads to symptoms that are pretty similar to other encephalitis symptoms, but pretty mild, and you can get rid of it, and most people don't have symptoms, but it can trigger Guillain-Barré, which is going to be a nerve-related dysfunctional disorder there, and seizures. It can cause issues with the baby if you are pregnant as well. We already talked about rabies, so I am not going to cover rabies because we covered it pretty well in that lecture. We already know 100% fatality if you have symptoms. Your only hope really is post-exposure therapy or vaccination beforehand. We also talked about polio, so I won't be going into that one, but oh my God, 72 years in an iron lung. I don't know if you guys really quite appreciate what an iron lung is.
Yeah,
Your
you're
body
stuck in there.
inside of an actual encased, like it looks like you're going into a hyperbaric chamber, like for your body, but your head is out. And you can have sometimes your arms out too and function that way, but like typically like it's causing pressure on the volume of your body, like your fluids in your body and also the musculature in your body to force your body to breathe because your actual diaphragm cannot breathe for you because you're paralyzed.
Did
Yes.
you know about Paul Alexander before?
I didn't know that he was there for that long.
Yeah.
Yeah,
Is there like a
72.
movie?
Oh, no, they
Maybe.
didn't. The Iron Lung movie being made by Markiplier.
It's not the,
Okay. And you can agree with that, right?
I think it's pretty good. I don't think it's the best.
It's one of the best. It's
Anyways, it's good. Anyways, everybody who wants to talk about horror games, you can come talk to me in Grace because obviously we play horror games. But it was a super tiny virus. We actually had to create a way to filter it out of the water supply because it was becoming such an issue. And you see those big hallways of just iron lungs of kids and people in them for years and years because they're completely paralyzed and can't breathe for themselves. So those pressure devices are going to force you to breathe. You're lucky if you can come out of one alive. So we don't have those as much anymore. We have that in Pakistan, Afghanistan, just because contaminated water. So we're still putting forth vaccine efforts there. Super tiny virus, we had to learn how to filter it out of our water as part of the reason that and vaccination, why we don't have it here in the United States anymore. And because we had to have things in place to filter out one of the smallest viruses that we've ever known, which is the coronavirus that causes polio, we were able to help filter out COVID from our water once that started showing up in our water sources. So we also have polio to thank for making us safe again with COVID, giving us a chance. All right, tetanus and botulism are our last two. These are caused by bacteria Clostridium tetani and Clostridium botulinum. Tetanus, Clostridium tetani, obviously. Clostridium means it's endospore. If it's endospore, it is gram-positive. Those are things that we know about it just from the fact that it's Clostridium. Clostridium is also anaerobic. We learned about that from Clostridium perfringens, or gas gangrene pause. So Clostridium is anaerobic. It is gram-positive. It is endospore forming. And these things have a lot to do with the presentation of disease. Bacteria gets into your body. Those endospores go back into the vegetative form. They start making toxin. The toxin is what causes the disease, not the bacteria themselves. And if you can't vaccinate against the toxin can't do its job. And then the infection is pointless. It can't do its thing. So we vaccinate only against the toxin for this one. Tetanus toxin called tetanospasmin. Where is this? There it is. Causes a spastic paralysis. That means you will flex your muscles and they get stuck. So spastic paralysis, stuck. You can't relax. So no relaxing for the muscles. You die from asphyxiation because you suffocate, because you can't breathe anymore, because your diaphragm will also seize up in the spastic paralysis. Starts with lockjaw as your like one of your early signs early on in the disease. If somebody were kept in a hospital that had actual presentation of tetanus, which is pretty rare nowadays because of vaccination efforts, but there's not anything we can really do for you. You have to wait it out. They can give you antitoxins, but usually it's gonna have to run its course. So they'll put you in a dark room, really quiet, and keep you segregated from things that could stress you out and make you seize up, because then your muscles are gonna stay like that until the toxin works its way out of the system. So spastic paralysis, can't relax the muscles. Obviously, we know, and this can happen in third world countries because this endospore exists, we've already said, pretty much everywhere in the soil. You step on a nail, it gets introduced into your foot, you get tetanus. Well, give birth in a place where you don't have sanitary conditions, the baby gets exposed and leads to a neonatal version. So vaccination is essential. Botulism has a similar toxin that blocks the release of acetylcholine. So this is a trigger in your nerves that tells them to do things. It's a signal passed between nerves. So now we can start moving our muscles, we can start doing things, we can breathe, all of this. That signal can't get sent because botulinum toxin blocks that from being sent. This leads to a flaccid paralysis. So Clostridium tetani, we have spastic paralysis, can't relax. Clostridium botulinum with the botulinum toxin, you can't contract. So you're flaccid, typical normal paralysis that we would think of with paralysis. You can't contract. Great for put that toxin in your forehead and you can't use your forehead muscles anymore. But also if it's all over in your body, you can't breathe anymore because your diaphragm can't do things anymore. It's not capable of contracting and relaxing to help power your breathing. So same exact way to die. It's just one's from not being able to relax and the other one's not being able to contract. Make sense? All right. Oh my God, there's another thing. Okay, African sleeping sickness. This is a Trypanosoma brucei. We talk about Trypanosoma cruzi being associated with Chagas disease. We talk about in the blood infections, but sleeping sickness is going to associate with brain. It makes you just randomly kind of fall asleep and have other associations with that. It's a protozoal disease transmitted by the tsetse fly. So we don't have this here in the United States. This is an African illness. So you can like literally just like fall asleep, extreme fatigue. So you'll just be like falling asleep, walking around and stuff. This is not narcolepsy. It's caused by the effects of this illness affecting the brain function. It can also lead to behavioral changes and other sleep disturbances. You'll eventually die from coma or secondary infections or heart disease. There's our summary. We'll go ahead and go to lab. While you guys are packing up, I'll go over the questions. You don't have to answer them. What pathogen causes the deadliest form of meningitis? That was Neisseria meningitidis. That was a bacteria, it's gram-negative. Diplococcus. What organism is associated with, what type of organism is associated with meningoencephalitis? Is it a virus, bacteria, protozoa, or prion? Meningoencephalitis was a protozoa. Those are our brain-eating amoebas. What type of pathogen is the most common cause of encephalitis? So we're talking about acute encephalitis. Those are viruses. Those are arboviruses. These are common types of questions you'll see on the exam, by the way. I might ask you what type of pathogen, or what's the name of the pathogen, or this pathogen, what disease does it cause? There's so many freaking diseases, you'll get a lot of general questions like that. You'll have multiple choice though, so it should be easier. What type of paralysis does tetanus cause? That was spastic paralysis. And what pathogen causes botulism? That's Clostridium botulinum. Gram-positive, anaerobe, endospore forming. Flaccid paralysis.
My woke mind virus.